Hello, everyone. My name is Gokul, and with me, I've got Sophie as well. So, both of us today are going to be presenting a lecture on all the different special senses. So, if you click the next one, Sophie. Um, okay, yes. So, we're doing smell, taste, vision, or audition. So, this is all the stuff we're going to cover today. So, smell and taste are probably less likely to come up as essays in the actual exam. So I've reduced the amount of experimental evidence. You can still get some MCQ questions on it. And I'll kind of highlight which areas I think are more important, but also some of it is important for you to know in terms of your overall understanding of neuroscience. Then vision and audition are very kind of like common essay questions. So that side of the lecture is maybe like the more important stuff today, I think. But all of the four different special senses kind of have a lot of similarities to them, which we can talk about. And then using those common principles, you can understand the sort of big picture and put it together when you actually write your essays or answer your MCQ questions. So if you hit the next slide, Sophie. So the core of the sensory systems, and if you will, the central dogma of it, is that you have an initial signal. It doesn't matter what that signal is. It can be chemical, in cases of smell and taste. It can be light in terms of vision. It can be sound in terms of audition. Then after that, you have transduction of this signal at the level of the receptor. So basically, what that means is that you convert this initial signal, which so this essentially this energy form into an electrical signal that can then be transferred to the central nervous system. Um, so all of the different receptors, whether it be sort of the retinal photoreceptor cells in the eye, or whether it's an olfactory receptor picking up odorant molecules, what they're all of them are doing is converting this initial signal into electrical pathways by the opening of ion channels, converting them into action potentials, which can then be transferred to the brain as information. And then at the level of the brain and spinal cord, you have higher processing. So once you kind of understand that, we can think about what exactly goes on in each of the different sensory systems. So I'm going to start off by talking about chemosensation. So that involves gustation, which is taste, and olfaction, which is what I'm going to start off by talking about. So olfaction or smell basically occurs at the olfactory epithelium by the olfactory bipolar sensory neurons. Um, so in terms of where these neurons are, I think this image on the right makes it quite clear. So if you could just maybe hover over where the image is on the right, Sophie, just over where the olfactory epithelium is. Yeah, just where the nose is on the right as well. Sorry. I know that's, yeah, exactly. Yeah. So if that's basically where the actual olfactory epithelium sits. So that's the only part of the body that can actually detect different smells. So the olfactory receptor neurons have like, they're ciliated. So they have these small hairs and humans have more than 400 different types of receptor neurons. So even though we have so many variations in the actual exact type of olfactory neuron, all of them seem to have a kind of common, pretty stereotyped pathway. So the pathway involves an odorant molecule dissolving in mucus, then activating a GPCR on the olfactory receptor neuron, which kind of activates your GS pathway, leading to the production of cyclic AMP. So the cyclic AMP then activates a cyclic nucleotide gated channel. So this then leads to an influx of sodium and potassium, leading to depolarization of the cell. Um, so the calcium, once it comes in, can then essentially negatively regulate the olfactory neuron. So it goes on to form calcium and calmodulin, and then that goes on to activate the calmodulin kinase, which leads to phosphorylation and inactivation of your initial C and G channel. You've also got an NCX exchanger as well, which is pumping out the calcium ions out of the neuron. So the negative regulation part is basically allowing the olfactory neuron to not be repeatedly activated when you have a smell for a long period of time. So you can kind of do this yourself. If you're in the kitchen, you often don't notice that you've got like a particularly strong smell. And if someone else walks in, they'll be like, oh, you've got a really strong smell going on. And the reason for that is because of this process where over a period of time, this negative regulation means that we stop noticing a cell and you don't get excessive stimulation by one specific type of odorant. Even though each neuron only has one odorant receptor, the key part of how olfaction works is combinatorial coding. So one odorant can actually activate more than one type of receptor. So if you go on to the next slide, Sophie. Um, so here we've got 14 different types of odorant receptors. So this is just an example. In reality, as I said before, we have over 400. But what you can see from this image is how any given odorant can actually activate multiple ones. So the reason that we have this combinatorial coding is that you have infinitely more possibilities of the kind of smells that we can perceive. So if each smell was only able to activate one receptor, we could only smell 14 different smells. But because each odorant is able to activate more than one potential receptor, we can actually smell we can smell 2 to the power of 14. So that means if we only had 14 receptors, that would be over 16,000 combinations compared to just 14. So right now, we don't even know how many different smells we can actually perceive. So different people suggest that it might be over 1 trillion different smells. So that kind of tells you how many possible combinations of smells there are based on the system. So if you go on to the next slide, Sophie. So in terms of the actual olfactory pathway, so this is, I think, quite important for the exam. So this is more likely to come up in part A. So this is like a slide for you guys to flag and remember. So the axons of the olfactory neurons are unmyelinated. So they then penetrate the cribriform plate. So once again, I've got the image on the right, which kind of is a good overview of how the overall olfactory pathway looks. So the cribriform plate is basically a part of the ethmoid bone. So you can see that on the image of the skull that I have on the right as well. So collectively, these different unmyelinated neurons form the olfactory nerve. So these neurons are constantly dying and are replaced. So they get replaced over a roughly a 4 to 8 week period. So not that many neurons in the brain are capable of regeneration, but the olfactory neurons are capable of this. So once you have the neurons passing through the cribriform plate and into the olfactory bulb, we have to remember the olfactory bulb is laminated with 5 key layers. So if you just click the next one, Sophie, there should be another image that pops up. Yeah, so there's 5 key layers. There's the olfactory neuronal layer, the glomerular layer, the external plexiform, the mitral layer, and the granular cell layers. So this is at the level of the olfactory bulb, to be clear. So once the olfactory neuron comes, it synapses onto the mitral cells. So the mitral cell is the main important cell in the process of carrying these olfactory signals into the brain from the olfactory bulb. So using the different combinations of activation of these mitral cells, the initial sensation of smell is now converted into a spatial map. So different mitral cells, based on which receptors are activated, essentially, based on that, you can determine what kind of smell a person is smelling. So the mitral cells then carry these signals for higher processing. So an important thing to note in olfaction is that there's no thalamic relay. So unlike something like touch or pain or vision or taste, vision or taste or something like that, the thalamus is not involved in this process. The olfactory bulb does the process instead. So apart from that, it's also important that you guys are aware of modulation that the mitral cell is receiving. So you have a GABAergic input from the granule cells. You also have periglomerular cells that can also contribute to inhibition of the mitral signal. So the reason for this is that it's basically lateral inhibition, which you have in the spinal cord as well. So what you don't want is for your mitral cell to get activated by any small smell that you're perceiving. So you want it to only be activated when the signal is quite robust. As a result, you have this kind of lateral inhibition that dampens the network altogether, so you ignore irrelevant olfactory information. Yeah, so that's what's happening at the level of the olfactory bulb. So the mitral cell then goes onto the actual brain itself. So if you go on to the next slide, Sophie. Yeah, so it then carries the signals via the olfactory tract. So the olfactory tract divides into a medial and lateral olfactory pathways. So the medial tract projects to the anterior commissure. So a commissure is basically a way for signals to get from the right side of the brain to the left side or vice versa. So it's essentially it's a pathway that allows the hemispheres of the brain to crisscross. So you've got the anterior commissure, the posterior commissure, and the corpus callosum. Those are the sort of 3 really important ways that signals can get across from one hemisphere to the other. You do have a few other small commissures as well, but those are the 3 ones that I think are most important for you guys to be aware of for second year. So apart from that, so then the lateral olfactory tract, on the other hand, goes and runs into the olfactory cortex. So the olfactory cortex is not one structure, but it's actually like kind of a loose area of the cerebral cortex. So it usually refers to the temporal lobe. When we talk about the olfactory cortex, we're typically talking about the piriform cortex, the amygdala, and the entorhinal cortex. Um, So those different parts of the brain are basically involved in slightly different processes. The entorhinal cortex is part of the hippocampus, so that's important in memory formation. Exactly what the role of these different signals is and why the amygdala is involved and stuff like that is still kind of poorly understood. But we know that they go there, and we know the rough function of the amygdala, for example. So the amygdala is involved in fear. The entorhinal cortex is an important part of the hippocampus involved in memory formation. So maybe these structures in the brain are related to olfaction in a unique way that we don't fully understand yet. So then after the initial olfactory cortex, from there, the output can go on to other parts of the brain, including the orbitofrontal cortex of the PFC, the hypothalamus, and the brainstem. So the secondary pathways basically control things like salivation and appetite regulation and how we perceive food and our control of food. So, for example, in conditions with eating disorders, for example, we know that the orbitofrontal cortex of the PFC is often like disturbed and things like that, and they have different activities going on there. So, it might be that these are really important signals that control our overall perception of food and how we react to different smells, basically. So if you go on to the next slide, so the next slide is more just like kind of yeah. So this one is just more like kind of putting together what I've kind of already talked about. It just basically summarizes all the stuff about the olfactory pathways, where they're going, and kind of just makes all the information a bit clearer, I think. I think they're just useful diagrams to have. This is also like stuff that could come up in your MCQs, so it's important to have some awareness of it. So now we're going to talk about gustation, so the other side of sort of chemosensation. So the taste buds and the tongue. So the taste receptors are basically clustered on mainly the tongue, but they are also found on the palate, which is basically the back of your throat, the pharynx, which is kind of a different part of the back of your throat, slightly lower down. The epiglottis, which is the bit that covers your windpipe when you're trying to swallow, and the upper esophagus. So taste buds themselves come in three different forms, based on the sort of structure and the how it kind of looks basically under a microscope. So you have fungiform, foliate, and circumvolate, and each of these vary in distribution as well as their structure. So the images on the right kind of make it quite clear, which ones are which. So the most important ones, which cover the most of the tongue, is the fungiform, I think. So you've also got filiform ones that are involved in tactile sensation. So each taste bud has about 100 different taste receptors. They contact whatever you're tasting via the taste pore. They also have basal cells at the bottom, which may be stem cells, but we're still not 100% sure. And taste basically occurs when you eat something or you put something in your mouth. It dissolves in the chemicals in the saliva, and then it activates these different receptors. So if you look at the right image, the bottom right image, can you see the bit where it says taste test, taste pause? Could you maybe hover over that, Sophie, just to make that clear? Yeah, exactly. So that's basically like a loose idea for you guys to understand how a taste bud looks. So that's kind of the structure that you're thinking about. So you've got the basal cells at the bottom, you've got your gustatory cells running along it, and then you've got your taste pores at the top with your taste hairs. So food contacts your gustatory cells once it's dissolved in saliva at the top, and then it can activate these different cells. So if you go on to the next slide, Sophie. So the next part is for us to think about how taste is encoded. I think this is something that, well, to be fair, the slide before can also come up in MCQs, but this is also something that's like quite important as well to think about, which is how actually taste is encoded. So we've got sort of 5 core taste things. A lot of you might have heard of it sort of just in like colloquial things. So we've got salt, sour, sweet, umami, and bitter. So sweet, umami, and bitter are all kind of activated by a very similar pathway, which involves the influx of calcium through a voltage-gated calcium channel based on the activation of an alpha-gustusin, which activates PLC and mobilizes internal calcium stores as well. So the important thing for us to remember in relation to this process is that it's only these 3 that are activated with the GPCRs. The salt and sour are both ion-gated channels. So I think like an MCQ question that can definitely come up would be, which one of these 4 is activated by an ion channel or something like that. So it's important that you guys are aware in order to pick out what is what. So the images on the right kind of make these pathways clear. There is subtle variations between sweet, umami, and bitter in terms of the exact nature of the secondary signals, but I don't think this is going to be an essay topic. It's unlikely to come up as an essay, so I wouldn't really focus too much on it. But as I said before, it's important that you guys have a basic awareness of what's going on. So once you have the initial activation of these signals, neurotransmitters are released, and then they're kind of transmitted by the labeled line hypothesis. I'm not going to labor this point too much because, as I said, if it's not going to come up in the essays, I don't think we need to like focus too much on the experimental evidence. But it's there for sort of completion and just interest as well. So if you go on to the next slide, Sophie. So then once you've got the initial sort of signal that's been activated in the tongue, once the taste receptor synapse with a primary sensory axon, they kind of run to the medulla, they run to the solitary tract, so they run to the NTS in the medulla. So three cranial nerves are involved in kind of transmitting taste signals. So you've got the facial nerve, which does the anterior 2/3 of the tongue. You've got the glossopharyngeal, which does the posterior 1/3 of the tongue. And then you've got the vagus, which does the esophagus. So the more important ones for taste is obviously the ones on the tongue, because that's where most of the taste receptors are. So once you've got your primary neurons running to the medulla, the secondary neurons then run from the medulla to the ventral posterior medial complex of the thalamus. And then from the thalamus, the third order neurons run from to the gustatory cortex. So this is basically refers to the insula, which is basically the bit that's tucked under the sort of the temporal lobe and the frontal lobe, as well as the frontal operculum, which is basically like a part of the frontal lobe. I think the next slide has a nice image. But this is the overall kind of pathway of the gustatory system. So if you go on to the next slide, Sophie. This is just kind of like a summary and some useful images, I think, that kind of explain the anatomy as well as exactly what's going on in relation to the signal transduction stuff as well. So I think that's kind of that's sort of the all I have to say on taste and smell. Hopefully that's kind of answered any questions you might have and given you a pretty good rundown of what's going on. Does anyone have any questions at all? I mean, we can take more at the end as well, but if anyone has anything before Sophie starts talking about vision and audition, maybe we can just answer them early. Okay, if everyone's happy, then I think you can take over from me, Sophie, if you're happy to do that.
Sure, sounds good. Thank you for that intro to taste and smell. So next we're going to go on to talk about vision and audition, which are two slightly bigger topics, I guess, on your syllabus. It's quite a lot to get through in 40 minutes, so I've tried to concentrate on the main things I think are important. I haven't got loads of experimental evidence and things here, but I think, well, both are more fair game for essay questions, so that might be something you want to just look back on, but this should be a good revision of all the main points. So we'll start with vision, and just to kind of ease us in, we're going to have a quick reminder of the structures of the eye. So this is a nice diagram of the eye, and you can see the yellow arrows, which is the direction of light travel. So you go through the cornea, through the pupil, through the lens, and then light shines onto the retina. And this little bit in the corner here shows the structure of the retina. So it's kind of got this inside out structure where actually your photoreceptors, so your cones and rods, which is where transduction of the light signal occurs, are right at the back. And then the signal's transferred to the bipolar cells and then to the ganglion cells, which transmit information via the optic nerve to the brain. And in terms of focusing of light, the two main structures that you want to think about are the cornea and the lens. So the cornea actually accounts for about 2/3 of the focusing power of the eye, but that's non-adjustable, whereas the lens accounts for about 1/3 of the focusing power of the eye, but that's adjustable with your accommodation reflex. So the shape of the lens changes, and that helps you focus on kind of near or far vision. Okay, so if we move on to kind of the sensory transduction. So as I mentioned, we have photoreceptors right at the back of the eye, and these are the rods and the cones. And the cones can be further split into short, medium, and long wavelengths, depending kind of the optimum wavelength of light that they are sensitive to. And we have these two different types because they are used for different functions. So rods are used more in kind of dark vision. They're very sensitive to low light levels, whereas your cones are used more in color vision, and that's why you've got the different wavelength sensitivities. In terms of their distribution throughout the retina, this also links to the two different functions. So your rods are much more kind of peripherally located throughout the periphery, whereas the cones are more concentrated in the center of the retina, and particular in the fovea. And the fovea is, if we go back to this diagram, it's right in the center of the retina here, this kind of in dimpling. And that corresponds to the center of your gaze, that you direct towards whatever you're looking at, the object of your attention. And it's the area of our highest visual acuity. And that acuity is enhanced by an absence of blood vessels in front of that area of the retina, and also by the retinal nerve cell bodies being kind of pushed out the way, so a direct path of light can land on the fovea. So if we talk a bit more about the phototransduction process now, you get your light shining into the eye onto the retina, and that's absorbed by the photopigment in the rod or cone cell. And that causes photoisomerization of a molecule called retinal, which basically just means that it switches from its cis or trans isomer, or vice versa. And that switch triggers a conformational change in another protein called opsin, which diffuses within the disc membrane. So the photopigment's found in these little discs, which you can kind of see in this diagram here. And the opsin diffuses in the disc membrane and binds to and activates a G protein called transducin. And transducin goes on to activate phosphodiesterase, which breaks down cyclic GMP. And so you therefore have reduced cyclic GMP channels, and that actually causes closure of sodium channels and hyperpolarization of the cell. So it's a bit different to some of the other sensory systems, which, like, traditionally you get depolarization of the cell. So that's quite an important point to remember. And this hyperpolarization occurs in a graded manner to light stimulus. So the resting membrane potential of a photoreceptor, when it's fully dark, is about minus 60 millivolts. And then as the intensity of light increases, the resting membrane potential is the cell is hyperpolarized, and the resting membrane potential becomes less and less negative. And when the cell is fully saturated, then it's about minus 40 millivolts. And this, again, rods and cones are different. So where rods are much more sensitive to light, they saturate or bleach at ambient light levels. So kind of at relatively normal light levels, they'll be at their minus 40 millivolts and they'll be fully hyperpolarized. Whereas cones are less sensitive, which means even in bright sunshine, you won't have quite reached your minus 40 millivolts. So there's still some graded change. And if we just look at this diagram on the right, you can see some of the kind of molecular processes going on in that phototransduction pathway and the difference in the rod and cone responses. Okay, so there's processing at various stages of the visual system, and the first level of processing is within the retina itself. And you can see in this diagram on the right kind of the different layers of the retinal cells, like we talked about earlier. And the first thing I think it's important to note is the circuitry for the cones and rod photoreceptors, which differs slightly. So for cones, in the peripheral retina, the cones are slightly further apart from each other, and there's convergence of information, which means kind of two cones might connect to one bipolar cell, so the information from both cones converges. Whereas in the fovea, because it's really important that we have this high visual acuity, there's very little spacing between the cone photoreceptors in the retina, and there's a one-to-one relationship between cone and bipolar cell and ganglion cell. So information from just one cone goes to one bipolar cell, and that gives us really high acuity, but it does give us slightly less sensitivity to light because you need more light essentially to activate the bipolar cell. In contrast to this, the rods, you have like 15 to 30 rod cells that will converge on one bipolar cell, and that allows quite little light to actually still cause an impulse in a bipolar cell. So that allows this high sensitivity, but the acuity might not be as good because you don't know exactly where that light has shone on the retina, if that makes sense. And you also have these cells called amacrine cells, which you can see here. There are a few different types of amacrine cell, but this is in particular is an amacrine A2 cell, and that will allow the rod signal to piggyback onto a cone pathway. So the rod bipolar cell is connected to the cone bipolar cell, which then goes on to a ganglion cell. And if I just quickly go to the next slide, this is illustrating some of the cone circuitry within the retina. Okay, so as I mentioned, the next type of cell in this kind of processing pathway are the bipolar cells. And the way these are set up is because the visual system is more sensitive to local contrast or to edges rather than uniform light or dark per se. So the receptive field of the bipolar cells can be split into on-center or off-center. And what this means is on-center cells respond best when there is light in the center of their receptive field and dark in the periphery, whereas off-center bipolar cells respond maximally to dark in the center of their receptive field and light in the periphery. And this setup is enabled by them expressing different types of receptors on their dendrites. So the on-center cells express metabotropic glutamate receptors called mGluR6, and that means that they are depolarized in the light because when it's dark, the photoreceptor is releasing more glutamate because it's depolarized rather than hyperpolarized. So in the dark, when the photoreceptor is releasing more glutamate onto the dendrites of the on-center cell, you get activation of these mGluR6 receptors, which causes activation of a G protein closing cation channels on the on-center cell, whereas the off-center cells express AMPA receptors on their dendrites. So when you get hyperpolarization in the light and less release of neurotransmitter onto retinal ganglion cells. So I'll show you a diagram in a minute that brings all this together because I know that's a lot of information. But the final thing to say is you also have lateral inhibition, a bit like what Gokul was describing earlier, which helps to set up these on-off-center or antagonistic receptive fields. So I hope that makes sense. I know it's a lot, but the signal from the bipolar cells then goes on to the retinal ganglion cells, which also have these on and off center receptive fields. And we'll go into this on the next slide, but the retinal ganglion cells are also the start of what's called parallel processing in the visual system, which is essentially where different types of information are kept separate as the information moves up through the CNS pathways. So if we just look at this diagram here, this details the kind of on and off center receptive fields and how these are set up. So this is particularly with the cone cells. You've got the on-center cone cell, it's hyperpolarized, glutamate's released, and the on-center bipolar cell is depolarized, so neurotransmitter release is increased, and that's because of the metabotropic glutamate receptors, whereas the off-center bipolar cell is hyperpolarized, so transmitter release is decreased, and therefore the off-center ganglion cell firing is decreased. Whereas your cone in the surround of the receptive field, if that's in the dark, then your cone is depolarized, so you have more glutamate release onto the horizontal cell, which then acts to inhibit the bipolar cell. And then you kind of get the opposite situation with the off-center receptive field. So it might be useful to come back to these diagrams to just look at the setup of the on and off center receptive fields again, because that's a theme that comes up throughout the visual pathways in the visual system. So if we go into a bit more detail about these retinal ganglion cells, as I said before, there are several types of retinal ganglion cell within the visual system, and actually, in fact, more are kind of being discovered. And they now think there's kind of, I think, 30 plus types of retinal ganglion cell. But for our purposes, there are two well-defined retinal ganglion cells that you need to know about. The M cells, so the midget cells, and the P cells, which are the parasol cells. Actually, sorry, it's really confusing. The M cells are the parasol cells. They look like parasols, and the P cells are the midget cells, really helpfully. There are also a few other types of retinal ganglion cells it's just worth mentioning. So there are photosensitive retinal ganglion cells, and these are used to help entrain your circadian rhythms. So they go off to a different area of the brain to facilitate that. There are also non-M, non-P type cells, which have a different role in colour sensitivity, which we'll go on to talk about. And there are cells that are important for saccadic eye movements. So they're important in some of your reflexes, and they go off to somewhere called the inferior colliculus in the midbrain. To help control that. But for now, we'll focus on the M cells, the P cells, and the non-M, non-P type cells, which go on to kind of the visual cortex through CNS pathways. So if we compare the M and P cells, this, as I say, is the start of kind of parallel processing within the visual system. So the two types of cell extract different information from a visual scene, essentially. So the P cells have much smaller receptive fields than the M cells, so they're generally better able to discern detail within a scene. And the P cell axons conduct impulses much more slowly than M cells. So M cells would be more sensitive to kind of a fast-moving stimulus, for example. Similar to this, P cells' responses to stimuli can be sustained, whereas M cells tend to be much more transient. So again, useful for detecting motion. P cells are sensitive to the colour of a stimulus, whereas M cells are not. So again, if we're thinking about detailed discrimination, then colour discrimination is quite useful. And we'll go on to talk about how are able to discriminate colour. And then M cells, by contrast, are much more sensitive to low contrast black and white stimuli. So on this concept of parallel processing, it's seen throughout the higher visual system. Different types of information are processed and kept separate in different pathways. So as I've said, M cells are highly sensitive to low contrast stimuli and rapid movement, whereas P cells allow the detection of visual signals that relate to fine detail, including colour and a kind of high contrast. And on the topic of colour, P cells can do this because the receptive fields of the P-type ganglion cells can show red-green colour opponency, which is kind of another level on top of this on-centre, off-centre idea. And you can see a diagram of this just on the right. So you'll have red centre on, green surround off, red centre off, green surround on, and then the other way as well. So there are 4 different types of red-green colour opponent receptive fields. Also, this is where the non-M, non-P type cells come in. So they can show blue-yellow colour opponency. And essentially these colour opponency, the way that it's set up means that you can get comparison of the activity of different types of cones, so those 3 wavelength cones, at the same time. So that allows extraction of colour information through the whole spectrum of wavelengths, not just the individual kind of wavelengths that the cones are most sensitive to. So you need the opponency mechanism to be able to extract the range of colour information from a scene. Okay. So the fibres, the afferent fibres of the retinal ganglion cells go on to form the optic nerve, and that's what transmits information from the retina to the brain. So you can see on this diagram here, the optic nerves are exiting the eye, and then the optic chiasm is this crossing point in the middle, and that's the site of decussation of fibres from the nasal retina of each eye. And that crossing means from this point onwards, fibres of one side on one side of the brain are carrying information from the contralateral visual field. So if it's on the right side of my brain, it's those fibres are carrying information from what I see on to my left in front of me. Whereas before that, you've got both visual fields represented on each, if that makes sense. So yeah, they decussate at the optic chiasm, and then we get to the lateral geniculate nucleus, which is the thalamic relay of the visual system. So Gokul mentioned the olfactory system doesn't have a thalamic relay, but that's quite unusual for a sensory system. Whereas this is much more in keeping with other sensory systems, for example, the somatosensory system. And just like our retinal ganglion cells, in the LGN, magnocellular, parvocellular information is still kept separate. So each layer of the LGN, as you can see here, there are 6 different layers, and this is it stained nicely on a slide, gets a retinotopic input from just one eye, and that alternates between ipsilateral and contralateral eye. Retinotopic essentially means that the organization is the same as the retina, so the map is the same. Topic tends to mean map. And the electrophysiological properties of the LGN cells are similar to the ganglion cells that supplied them. So you get these on and off center cells, and you get the color opponent cells as well. And we can see this parallel processing theme come up again. And the selective lesion damage to the particular layers of the LGN helped to elucidate function. So those were the initial studies that helped us to work out what was going on and the structure of these layers. So the parvocellular stream of the LGN seems to be important for spatial resolution, the size of objects, shape, colour, whereas the magnocellular stream is important for high temporal resolution, so the location, speed, direction, particularly of a rapidly moving object. So yeah, as we can see here, the parvocellular layers of the LGN are layers 3 to 6, the magnocellular layers are 1 and 2, and the coniocellular layers are ventral to each principal, either parvocellular or magnocellular layer. So they're these little pink bits in between the green and yellow layers. Okay, so from the LGN, we get to the primary visual cortex, or V1, and this also has a retinotopic organization. So again, things are represented in the same kind of place as they would be in the retina. So you have these stellate cells which receive the information from the LGN and they pass it to the dendrites of pyramidal cells for then the output to other areas, higher areas. And there are a few different kind of types of information and functions going on within V1. So first is binocularity. So you have these cortical columns in layer 4 which show that information from each eye in layer 4 of the of V1 is kept separate, but you then get convergence of the information via projections to the layers above and below, and the layers which have convergence or binocular information are really important because you need to kind of combine the inputs from both eye, which will be very slightly different, for perception of depth and distance, which is obviously very useful so we don't go bumping into things. There are also two primary types of cell in V1, simple and complex cells. So simple cells are found in layers 4 and 6, and these show orientation selectivity. So they will only fire for objects if they're in particular orientation. This is different to complex cells, which are found in layers 2, 3, and 5, and they have larger, more complex receptive fields than simple cells, and they give response to stimuli throughout the receptive field as long as they're in the correct orientation. Again, I'll keep harping on about it, but parallel processing pathways continue to be kept separate in V1. So you have different layers receiving the input from the LGN. So 4C alpha receives the magnocellular input, 4C beta, the parvocellular input, and 2 and 3, which receive the coniocellular input. And then you have these pathways in the within the primary visual cortex, which you can see in this diagram here. So there are two parvocellular pathways that you need to know about. The first one is the parvo-blob pathway, which is important for colour vision. So the blobs are essentially collections of cells that respond selectively to colour contrast, and they're centred on an ocular dominance column, which are these columns in layer 4, which are either from the ipsilateral or contralateral eye. And there are 4 classes of colour opponent cell with receptive fields tuned to colour contrast. So like we saw earlier, the colour opponent receptive fields. So you might have a cell that is excited and inhibited by green in the centre and inhibited by red in the receptive field centre. Or you might get one that's excited by red and inhibited by green in its centre. And once again, these do not respond strongly to either uniform illumination or achromatic contrast. It's particularly the colour opponency that we're looking for. and they're also not selective for orientation or direction. The other parvocellular pathway that we see is the parvo-interblob pathway, which is an interblob is literally an area between two blobs. And they are collections of neurons found surrounding the clusters of blobs, and they have complex cell receptive fields. So like we've gone into a bit earlier, and they show orientation selectivity, which is the necessary for the finer detail processing within the visual system. And these are binocular, so you get input from two eyes and they respond to achromatic contrast, but they're not sensitive to colour. And then in layer 4B, you have the magnocellular pathway, and this is mostly made up of simple cells, and you get both orientation selectivity but also direction sensitivity. And this is mostly based on binocular input, so input from both eyes. The cells in this pathway tend to have large receptive fields, and they're unable to discriminate the fine detail, but they're sensitive to low contrast achromatic stimuli. And this pathway also mediates visual attention and gaze via layer 5 outputs to something called the pulvinar thalamic nucleus for attention, and the superior colliculus for saccadic eye movement, so gaze. Okay. And now we move on to the extrastriate cortical areas. So this is the output from V1 to other areas. And once again, we're sticking with our parallel processing, and it's split into a ventral stream and a dorsal stream. So the parvocellular pathway that we've been talking about goes into a ventral stream. So you get your layer 2 or 3 output that goes to areas V2 and V5 within the visual cortex, the wider visual cortex, and then that goes towards the inferotemporal cortex. And that is important, sometimes known as the Watt pathway. So it's important in object form and colour recognition. And it's implicated in your long-term visual memory, so your memory of the kind of visual scene. And within this pathway, there are specialized areas, including the fusiform face cortex in the inferotemporal cortex, and that is an area that responds specifically to human faces, but not any other stimuli. So it's quite an interesting, really specialized area of the human brain. The other stream, the magnocellular stream, is your Ware pathway or your dorsal stream. And that's your layer 4B in V1 goes to V2 and V4 within the wider visual cortex, and then to your medial temporal lobe and posterior parietal cortex. And this is important for motion recognition, and it goes towards, understandably, areas for sensory motor coordination, so kind of responding to your this moving object by things like reaching and grasping, directing those kind of movements. And unlike the ventral stream, there's not much visual memory that is associated with this pathway. It's just in your very short-term memory so that you can respond to what's going on. Okay, so that was vision. Does anyone have any questions on that so far? I'll try to answer. Okay, if not, we will move on to audition for now. So this is a quick reminder of the structure of the ear, which you have your external ear, your middle ear, and your inner ear. And if we go right back to basics, it's important to remember that a sound wave, which is the stimulus that we want to sense, is a pressure wave of alternating compressions and rarefactions of the air. So the external ear has a few different functions. You get resonance within the external ear and the ear canal, which leads to an increase in pressure of the original sound. And so you get an increase in amplitude. And this amplitude modification also depends slightly on the frequency and location of your sound source. So you're already starting to get some information for sound localization when that is processed a bit higher up in the pathway. As I said, you have your auricle, your external ear, and then that goes into the ear canal, and that finishes at the tympanic membrane, also called the eardrum, more colloquially. And that takes us on to the middle ear, which is an air-filled pouch connected to the pharynx by the eustachian tube. And this eustachian tube allows pressure equalization between the pharynx and the middle ear, which is quite useful on things like planes, and when there's big pressure differences, then that allows equalization, which is quite important. But in hearing specifically, the middle ear connects the tympanic membrane to the oval window by these ossicles, which are these three tiny bones, the malleus, the incus, and the stapes. And sound goes in that direction, so from the malleus to the incus to the stapes. And you essentially get vibration of the tympanic membrane. And those vibrations are transmitted via the ossicles. And they match the impedance of the air to that of the fluid within the cochlea, so within the inner ear, to prevent any loss of sound pressure, so sound amplitude. You also get actually some magnification of the sound pressure because the eardrum surface area, so this tympanic membrane here, is about 20 times larger than the stapes footplate here. So you get magnification of the pressure that's then transmitted to the cochlea or your inner ear. And at the start of the inner ear here is the oval window. So the inner ear is made up of your cochlea, which is your hearing organ, and that's what we'll talk mostly about today, but also your semicircular canals and otolith organs, which are important for balance. They make up part of the vestibular system. But if we think about the cochlea specifically, it is a coiled structure of progressively diminishing diameter, and its interior consists of three parallel fluid compartments, which you can see here. So we have the scala vestibuli, scala tympani, and scala media in the middle. And the oval window, so this one attached to stapes, is at the basal end of the scala vestibuli, and the round window, so here, is at the basal end of the scala tympani. And the organ of Corti, which is where sound transduction occurs, sits within the scala media on top of the basilar membrane, which you will see in the next slide. So this is the basilar membrane within the scala media, scala tympani here, scala vestibuli here. And it's useful to just look at these diagrams in different kind of planes to get a sense of the cross-sectional area of the cochlea and where you're looking, because it can be a little bit confusing. So if we think about auditory transduction or transduction of sound signals, it is enabled vibrations of this tympanic membrane, which are then transmitted by the ossicles in the middle ear to the oval window of the cochlea. So kind of what we were talking about just a minute ago. And you get corresponding movement when that oval window vibrates. You get corresponding movement of the round window, kind of around the semicircle of the cochlea. And that's because of the incompressibility of the liquid within the scala compartments. And you get these vibrations essentially causes a pressure gradient to be set up between the scala vestibuli and tympani, which causes this basilar membrane here and here to move up and down. When that happens, there's a shearing force that is generated between these hair cells, which are these cells here, lining the basilar membrane, and the overlying tectorial membrane. So tectorium literally means tent, overlying. And that causes these little stereocilia, so tiny hairs on top of the hair cells, to be deflected. So when the stereocilia bundle is deflected in the direction of the tallest stereocilium, you get opening of transduction channels at the tip of these cells, so little ion channels, whereas when the bundle moves in the opposite direction, you get closure of these transduction channels. Opening of the transduction channels allows entry of potassium ions, so they're potassium ion channels, which causes hair cell depolarization and calcium entry, causing release of excitatory neurotransmitter from the basal region of the hair cell onto fibers of the auditory nerve, which is one of our cranial nerves that goes up to the brain and continues to transmit sound signals up those CNS pathways. Like in the visual system, you get graded changes in the hair cell's receptor potential. So that causes a graded change in the amount of neurotransmitter released onto the auditory nerve fibers. And this kind of system works both ways, because at rest, when there's no movement of the stereocilia, about 50% of the channels involved in stimulus transduction are open. So when it's deflected either way, you get a change in kind of transduction and transmitter release. Okay, so if we think about what's going on within the cochlea itself, within the cochlea itself, the main thing that's going on is analysis or extraction of the frequency of a sound. And this is because of quite a clever setup of the basilar membrane and its mechanical properties. So different audible frequencies of sound induce motion of the basilar membrane at different points along its length. So you get your vibrations, sound vibrations, which produce a travelling wave of displacement of the basilar membrane from the base to the apex. But for different frequencies, you will get peak amplitude of this wave, or peak amplitude of basilar membrane movement at different points. So for high frequencies, the peak amplitude of the wave occurs near the base, and for lower frequencies, this shifts progressively towards the apex. And that's because the basilar membrane increases in width and decreases in stiffness from base to apex. So you then have your hair cells, which are the like transduction cell. And interestingly, you have inner and outer hair cells, which I'll show a diagram of in a minute. But the inner hair cells are the important cell for transduction itself, and they receive about 90 to 95% of the afferent myelinated auditory nerve fibers. So each inner hair cell is actually innervated by about 10 auditory nerve fibers. So you get divergence in output from the cochlea. So one inner hair cell will activate 10 fibers, which each carry its own signal. So there used to be a bit of a question about, okay, well, why are there all these outer hair cells? Because there are actually about 3 outer hair cells for every inner hair cell. So what's going on there? And they are thought to have a role in amplification of basilar membrane motion. So they have a property called electromotility, which means the cell body actually shortens up by up to several micrometers when depolarized and elongates when hyperpolarized, so depending on those opening of those transduction channels. And that allows them to move, so electromotility, and amplifies the motion of the basilar membrane. And then, as we've said, depolarization of inner hair cell causes activation of auditory nerve fibers. And the auditory nerve fibers have a characteristic frequency to which they are most sensitive, And then their thresholds increase as the frequency moves away from this value. So if the sound wasn't quite at the characteristic frequency of a particular auditory nerve cell, then the intensity of the sound would have to be higher to create the same response in that auditory nerve cell. So you get these V-shaped tuning curves, which you can see on this diagram here. So different points along the basilar membrane, innervated by different auditory nerve fibers, will have different tuning curves. And yeah, the characteristic, and that's known as a tonotopic organization. So once again, the characteristic frequency of an auditory nerve fiber is determined by the point of innervation along the basilar membrane. But as I've said, you've also got some intensity tuning. So with increased sound intensity, you will get increased firing rates of the kind of auditory nerve cell up to a saturation of a certain value, about 30 to 50 decibels. So let's just see if we've got a diagram. No, sorry. Another thing that aids pitch discrimination or frequency discrimination within the cochlea for low frequency sounds up to about 3 hertz is something called phase locking, which means that auditory nerve fibers will trigger an action potential at the same point within the phase of the sound wave, which means that if we think of frequency as how often a sound wave is kind of reaching a peak or reaching a trough, you can measure it from any point, then the number of waves per second is encoded by the firing rate of the auditory nerve fibers responding to that sound. However, because of the refractory period of just any nerve cell, once you get above a certain frequency, those waves are coming so often that essentially the auditory nerve fiber can't keep up. So phase locking only works for low frequency sounds, but it's quite useful. So moving away from the cochlea, the auditory nerve fibers then come into the brainstem. And the first kind of junction we want to talk about is the medullary cochlear nucleus, which is the first site of neural inhibition. So similar to the visual system, which is arranged retinotopically throughout lots of the layers, the auditory system retains this tonotopic organization, and that is present in the medullary cochlear nucleus, and each neuron type within the cochlear nucleus has a distinct projection pattern to the higher auditory centers. And as a result, the auditory pathway comprises at least 4 parallel ascending pathways that simultaneously extract different types of acoustic information from the signals within the cochlear nerve. And this is the first site of neural inhibition within the auditory pathway. As we move up, we come onto the superior olivary complex, which is quite important for sound localization. And the superior olivary complex comprises of a medial superior olive and a lateral superior olive. And the medial superior olive compares interaural time difference, whereas the lateral superior olive compares interaural intensity difference. And these are useful for different types of frequency. So if we think about the pathway from the cochlear nuclei to the superior olive, the bushy cells of one cochlear nucleus actually project bilaterally to the superior olivary complex via 2 courses. So one side cochlear nucleus goes to both superior olivary complexes, one on each side. And one course through the medial superior olive compares, like we were saying, interaural time difference, so the time of arrival of the sounds at each of the ears. And and that's useful for the localization of low frequency sounds. So you get a map within the medial superior olive, which is generated of your interaural time differences, which is essentially the phase difference of two sound waves. And that's enabled by the phase locking phenomenon that we were talking earlier. So the firing of auditory nerves within one ear will be aligned to the phase of the sound wave when it reaches that ear. And there's been kind of debate about how is this encoded, like how do we compare the inputs from both ears. And one model that is seen in different animals, there's quite a lot of evidence for animals within, such as the barn owl, that there is a system called a Jeffress model, which basically suggests that there's an array of detectors. So I think I've got a diagram here. So if we look at this, you've got this array of detectors, which are essentially cells within the medial superior olive, and they detect coincident inputs from both ears. So the cell that will fire is the one where you get input from both ears at the same time. And the information for that is transmitted by delay lines. So these are the delay lines, comprised of axons with systematically differing lengths. So depending how far from each ear, how long the delay line is at the cell that fires, you can work out the difference between each ear. So if it's the same, then the distance or the phase must be the same at each ear because it's travelled the same distance. The signal has travelled the same distance from each ear, whereas as the difference increases, then the localization of the sound must be closer to one ear than the other. So yeah, interaural time difference delays increase systematically as the sound moves from the midline. So if the sound were directly in front or behind of you, towards the sign. Like I say, there has been evidence found for this in the barn owl. However, evidence is slightly more dodgy in humans, so it's worth looking at the evidence for that for your preclinical essays, because this could be a topic that comes up. The other cause from the medial cochlear nucleus travels through the medial nucleus of the trapezoid body to the lateral superior olive, and this compares interaural intensity differences. And this relies on inhibitory pathways as well as excitatory pathways. So you get excitation of the ipsilateral lateral superior olive, and the contralateral lateral superior olive is actually inhibited. And basically neurons within this complex integrate the ipsilateral excitation and the contralateral inhibition to measure the interaural intensity difference, and that helps to localize high frequency sounds. You get more of an intensity difference with high frequency sounds because sounds with wavelengths similar to or smaller than your head are deflected by the head. So you therefore get a decrease in intensity, and the intensity at the near ear tends to be greater than the intensity at the ear furthest away from the sound. So if the sound was here, I'd hear it louder than this ear Okay. So these auditory pathways from the olivary complexes and the cochlear nuclei converge at the inferior colliculus, and this information is then relayed to the medial geniculate nucleus of the thalamus, which is our thalamic relay for the auditory system, and information is passed on to the auditory cortex. So A1, the primary auditory cortex, has tonotopic organization as well as with our earlier levels of the auditory system, and basically generally audition is not as well defined as vision, but there is some evidence for clustering of neurons with similar properties. So the most neurons in the auditory cortex are preferentially driven by stimuli with greater spectral and temporal complexity than pure tones and broadband noises. So it seems to be like it's not just the neuron has its preferred frequency and that's it, as in lower down in the auditory system. You might get things like they have a preferential response latency or loudness or rate and direction of frequency modulation even, so they're responding to changes in frequency. But it remains to be seen how the various kind of maps intersect. And then if we think about further projections within the auditory system, again, like the visual system, there is some evidence for a dorsal and a ventral processing stream, but this hasn't been widely characterized yet and work is ongoing. And as with any of the other sensory systems, there are also reciprocal projections to lower areas within the CNS pathways for top-down feedback. So things like attention can be modulated, and you can tune out sounds that essentially you don't want to hear. So lots of forward and back signaling to create a very complex system. Okay, so that's all I had to say. This just shows that we've got the excitatory input in the medial superior olive and the inhibitory input in the lateral superior olive, which set up the measurement of interaural time and intensity differences. So yeah, two circuits set up differently for different purposes to extract localization information, but for different stimuli. And yeah, that is all I had to say. Let us know if there are any questions. You're welcome to email either me or Gokul about anything. And please also fill in the feedback form. There should